All right, so without any further delay, let's go ahead and jump in this morning. So on Wednesday, we had finished talking about the autonomic nervous system, and we had talked about the adrenal sympathetic pathway, and the thing that really I think the big thing that separates it from the other autonomic pathways that we looked at is that the neurotransmitters that the adrenal medulla secretes are dumped into the bloodstream. And as a result, as we described on Friday, because they are dumped into the bloodstream, those neurotransmitters aren't targeted towards a specific organ. They go everywhere, right? And that's why we see these kind of global sympathetic responses when we have an acutely stressful situation. Again, something like, almost getting into a car accident on Magnolia, or maybe you're out for a hike and you see a rattlesnake, or you're surprised by a rattlesnake or something like that, right? When those sorts of things happen, you have a much different response than kind of our normal day-to-day, minute-to-minute, sympathetic, parasympathetic maintenance of homeostasis, right? So what we have to do then is we have to start to consider these messengers that are being carried in the bloodstream. And that leads us into talking about the endocrine system, which is the last part of this unit, right? So the endocrine system is characterized by chemical messengers, hormones, being carried in the blood. Pull up my notes here really quickly. There we go. So hormones are chemical messengers that are carried in the bloodstream. I'm going to put this up for just a minute. I'm not quite ready for the screen to be down yet. There we go. So hormones in the endocrine system. Hormones are chemical messengers that are carried in the blood. Carried in the bloodstream. Okay, carried in the blood. Sorry about this. Okay, so and that's kind of where we're going to turn our attention to over the next couple of lectures, as I said. Sorry, got a little bit thrown off there. Okay, so chemical messengers carried in the bloodstream. There's a whole bunch of them that we're going to be talking about. We obviously aren't going to cover all of them. There are many, many more that we aren't going to cover. But we're going to talk about the major endocrine glands in the body, as well as the hormones that they secrete and their targets and things like that. One of the things that we do know about hormones is that they can have a really profound effect on our physiology. Even they can have a profound effect on our appearance as well as our behavior. The hormones were first discovered. The science of endocrinology is fairly old. They were first discovered in the mid-1800s. In 1849, there was a German physiologist named A.A. Berthold who had these roosters. If you've ever spent any time around roosters and chickens, you know that the roosters not only look different than the hens do, but they also act a lot differently as well. They can be quite aggressive in some cases. Obviously, not only towards the hens in terms of sexual advances, but they can even be aggressive towards intruders into their flock and things like that, right? Roosters often will have a really big red comb on top of their head. If you've never seen one in person, just think about the old Foghorn Leghorn cartoons, right? In fact, he's a pretty good example of a rooster in terms of the way that he strutted around the farmyard and things like that, right? So roosters not only act differently, they also look a little bit differently than the females, than the hens do. In addition to that big red comb, they usually have really brightly colored feathers as well, whereas the hens tend to be a little bit more dull in terms of their coloration. This really isn't an uncommon thing in birds. We see that the males usually are the really, really colorful ones, whereas the females are a little bit less colorful. Towards the end of the semester, when we talk about the reproductive system, we'll revisit that whole concept. But anyhow, this physiologist, the German physiologist, A.A. Berthold, had these immature roosters, and as they were developing, he castrated them, removed their testes. And what he noticed when he did that is that their behavior changed. They became way less aggressive. They weren't making the sexual advances towards the hens that they usually were. They became less aggressive towards him. But also their appearance changed as well. The coloration in their feathers was not as colorful as it would have been in an intact rooster, one that hadn't been castrated. That comb on top of their head kind of shrunk a little bit, and it was way less colorful than it was. So their appearance and their behavior changed. And then what he did was kind of an interesting thing. He actually transplanted testes back into the castrated roosters. And he noticed that when he did that, those traits came back, right? The comb grew back and it became a lot more colorful. The coloration in their feathers came back, and of course the aggressive behavior came back. I don't know about you, but if somebody castrated me and then transplanted testes back into me, I would probably be kind of aggressive as well. But that's beside the point. What he noticed when he did the initial castrations, and this is kind of the important part of the story, is when he did the initial castrations, and then even when he dissected the animals that he had transplanted testes back into, he noticed that the only thing that was really changed was the blood supply. He noticed that when he castrated the roosters and when he removed their testes, he didn't cut any nerves. He only cut blood vessels. And when he dissected the animals that had testes transplanted back into them, he saw that a blood supply had grown back around those testes, and the nerves remained relatively unchanged. So he guessed, and he was correct, that whatever chemical the testes were putting into the body to change their appearance and their behavior, that chemical had to be putting into the bloodstream. Okay, and as a result, the science of endocrinology was sort of born, right? We became aware that there were chemicals in our blood that changed our behavior, that changed our appearance, and now we know that these chemicals also are changing our internal physiology as well. Okay, so it's kind of an old science, way longer than we really had the capability of understanding molecular structure and things like that. We were pretty basic in terms of our understanding, but we've known for a very long time that these chemicals are in our blood and they change the way that we look and the way that we act. So these hormones are these chemical messengers in the blood, all right? One of the things that we're going to be looking at in this unit is we're going to be looking at the endocrine glands themselves. Okay? An endocrine gland is a cell or a group of cells that secretes these hormones into the blood. Okay, so we have endocrine glands. Endocrine glands secrete hormones into the blood. We're also going to be learning about the fact that all of the hormones that we study have particular target tissues. Okay? One of the things that's a little bit tricky with target tissues. One of the things that's a little bit tricky when we put a chemical messenger into the bloodstream is that it does go to every single cell in the body. When I'm using neurons to communicate with cells in the body, that neuron goes directly to that cell. The neurotransmitter is released directly onto that cell, and there's not really any, I guess for lack of a better term, slop to potentially stimulate other organs around, right? But with hormones, that blood is literally going everywhere in the body, and your blood vessels go to just about every single cell in your body. So how are we going to ensure that only certain target tissues respond to a stimulus? Well, they've got to have receptors. Okay? Receptors are usually proteins, and proteins, as we know, are determined by our genetics, right? So these specific target tissues are going to have receptors for our hormones. We'll talk about where those receptors are located here in just a little while, okay? But they've got to have the receptors in order to be able to respond to that stimulus, okay? And then the other thing that we'll talk about as well is we'll talk about how the activity of these hormones are terminated, okay? So the last question that we'll talk about here is how is a hormone's activity terminated? So we'll talk about that as we kind of go through here. So we have a couple of questions that we want to ask about. So again, hormones affect our appearance, they affect our behavior, they also affect our physiology, the way that our internal body sort of functions. And this is really just another way for us to have cells communicating with each other in the body, right? At the end of 50A, we started to talk about the nervous system. We've been talking about the nervous system more or less for the beginning quarter of this class as well, right? It's a big one with a lot of aspects to it. And the nervous system is a way for cells in our body to communicate with each other. The endocrine system is just another way for cells in our body to communicate with each other. We're just using a slightly different delivery method, right? So really we're kind of sticking with the theme here. It's worth noting that hormones aren't the only chemical messengers in biology. There's another class of chemical messengers that are really important for some animals. Those are pheromones. Okay? Pheromones, just as just kind of an important aside, pheromones are chemical messengers that mediate interactions between individuals. So hormones mediate interactions between cells within an individual. Pheromones mediate interactions between individuals. And they can be really important for a lot of species biology. Okay? Territory marking is an important one. If you have a dog, or maybe even a cat, and you take them for walks, one of the things that you might notice is that they pee an awful lot, probably more than seems reasonable, right? Why would an animal need to pee that much on a single walk? It doesn't matter that they have to urinate. There are chemicals in that urine, the pheromones, that are going to tell other animals that this is their territory, right? That territory has now been marked. So when the next dog or whatever comes along, it's going to sense those chemicals and be aware that there was another animal there, right? So territory marking is a really important thing. Another really obvious one is reproduction. Okay, animals will use pheromones for all sorts of different reproductive activities. Some of the more probably obvious ones are things like receptivity indication, knowing when another animal is ready to breed or mate, I guess I should say, is an important one. But heck, even gender recognition is important as well, right? Deer walking around the forest probably don't have the cognizance to look at each other and say, she's looking good, right? They just see another animal there, right? There need to be chemicals to tell them that this is a potential mate, right? So pheromones are responsible for gender recognition, sexual attractance, not only just receptivity indication, but also just general sex attractance. I have a funny story about that one. So this is a while ago. This is like 10 years ago or so. My wife's mom had been renting a place in Colton. And one night she was coming home from work, and she picked up this dog in the rain. And she was a really sweet dog. She was like, I don't know, probably about this big, probably like a German Shepherd something mix, I don't know. She was really sweet though. And she had her for about a year before we actually found somebody to adopt her. So she just kind of hung around. And even though my mother-in-law had her for about a year, we never really gave her a name. We just kind of called her Girl. And that was really about it. But she was really sweet and stuff, but she definitely had not been spayed. Okay, so it was like in the springtime at some point. Girl had gone into estrus. She'd gone into heat. And I was over at the house fixing some windows for my mother-in-law. Her landlord, wasn't doing it, and she needed them fixed and stuff. So I said, I'll just come do it. So I went over, and I was in her backyard working on these windows. And Girl's sacked out in the sun, like dogs tend to do, but she's in heat. But anyway, I go into the front yard for something, to go get something out of my car. So I go through the house and go out into the front yard, and I'm not making this up because I wouldn't lie to you guys. There were no fewer than 10 chihuahuas in the front yard, all trying to find a way into the backyard. And they had little tiny chihuahua erections. Like, they were ready to go, right? But it was crazy. Like, I'd never seen anything like it. But how did they know? Pheromones, right? Girl's sacked out, right? She's not actively trying to do anything except sleep, but she's in heat. She's giving off these chemicals, these pheromones, and all these little tiny chihuahuas are trying to get into the backyard to mate with her. And it was one of the craziest things I've ever seen. But they were like rabid little dogs. I was just like, wow, that's astounding. And later that afternoon, one actually did somehow get into the backyard. I don't know how it did, but I heard this yelping behind me, and this chihuahua's like just going to town on Girl. And I was like, no way, we aren't having puppies. And I like came up and I kicked the dog off of her. I was like, this is not happening on my watch. Anyway, it was crazy. I never did figure out how that chihuahua got into the backyard. But at any rate, I booted it and chucked it back over the fence, and I was like, get out of here. Little horn dog. Anyway, it was crazy. It was something else. But pheromones, okay. Parent-offspring interactions are another way that pheromones work, right? You probably have all seen that movie. What is it? Penguins, Happy Feet. Yeah. All the penguins are out on the beach, and the moms and babies or dads and babies have to find each other, right? I hate that movie, by the way. I detest that movie. But nevertheless, it's a good example of the babies and the parents trying to find each other. Again, these animals don't be like, oh, there's little Bobby. They don't do that. They have to have some sort of a chemical messenger that tells them that this baby is theirs. Okay? So parent-offspring interactions are important.
Why do you detest it?
I just don't like it. I think it's the animation that just creeps me out. It's like that other one, the Polar Express. Oh, yeah, like the animation in that one is just not right, man. But anyway.
Polar Express is a good movie.
No, the story's fine, but man, the animation is not okay.
Yeah.
There's something otherworldly about that. But anyway, parent-offspring interactions are another important thing. The bottom line is that pheromones are super important to a lot of animals' biology. They're probably very unimportant to our biology. Okay, as far as I'm aware, and I could be wrong, there aren't really any good, well-documented cases of pheromones doing much of anything at all in our biology. One of the things that was cited for a very long time as being attributed to pheromones is that we do know that when women who live in close proximity to each other for enough time will eventually start to realize that their menstrual cycles become synchronized with each other. This was first noted in sorority houses in the 1970s when you'd have like 30 or 40 women living all together. And give it 6 or 8 months, and all of a sudden you see these synchronized cycles. That was attributed for a really long time to pheromones. We know now that there's more to it than that, and it isn't really pheromones at all that's doing that. But that was attributed to pheromones for a very long time. But we know now that that's not necessarily the case. So there are not really anything in the human in the human world that really sort of does what pheromones do in other animals. I would imagine a lot of this has to do with the fact that we are, that we have things like language and stuff like that. We have words that we can use. We don't have to rely on chemicals to know who our kid is. Sometimes I wish we could, but we don't have to, right? Probably because we do have things like language and we have what we have as humans, it probably has sort of made pheromones relatively unimportant to our biology. So there's no proven sexual attractants or social indicators or anything like that. Occasionally you see them marketed, right? Like the cologne that will surely make you the life of the party or something like that. The reality is that if you want to meet somebody, just say hi to them. It tends to work a lot better. So anyway, a couple of different examples of chemical messengers, but we're focused on hormones, and these mediate interactions between cells in our bodies, and they are very, very important to our biology. All right. Okay, so let's go ahead and start to look at the chemical nature of hormones here. What are they made up of, and how do they work? That's kind of what we want to look at next. So I'm going to actually put my screen down. I'm going to need some stuff here in just a minute. So I'm going to put my screen down and start working around my screen here. Okay. Okay. So the chemical nature of hormones. Generally speaking, hormones fit in to one of 3 chemical classes. By far, the most common chemical class of hormones that we see are proteins. Okay? So the most common are protein hormones. Well, we'll talk a little bit more about their synthesis here in just a couple of minutes. I've got a couple of slides to show you. As you guys learned about in 50A, and probably as well in Bio 1, proteins can do a number of things in terms of their shape. Most of the proteins that we're dealing with in this instance are globular proteins, so they have this 3-dimensional structure to where that amino acid chain is kind of wound into a very, very specific 3-dimensional shape or something like that. So they typically are globular proteins. We'll talk about the significance of that when we start to talk about their interaction with receptors. Okay. Another class of hormones that we see are steroid hormones. Okay. Steroids are a class of hormone. These are derived from cholesterol. These are lipids. Okay. So these are lipids. We're looking at different kind of chemical behaviors here. These behave as lipids. They are lipids. And then our third and final kind of chemical classification of hormones is we have a third class called the amines. Amine hormones. Amine hormones get their name because they're derived from a single amino acid. Okay? might be saying to yourself, well, proteins are just a chain of amino acids. Why are you separating out the two things, right? Why am I listing the chain of amino acids, the protein hormones, different from the ones that are derived from a single amino acid? Well, the protein hormones largely are hydrophilic, meaning that they tend to dissolve really, really well in water. And that affects how they act on our cells. We're going to talk about that next. Steroids, as lipids, are exactly the opposite. We know that lipids are hydrophobic, right? So they don't dissolve well in water. That will affect how they act on the cell. Again, we'll talk about that in just a couple of minutes. Our amine hormones, it becomes a toss-up, right? It really depends on the amino acid that we're talking about. Some of our amino acids are hydrophilic, some of our amino acids are hydrophobic, so they don't fit very neatly into a box. So I'm separating them out because their behavior differs depending on the amino acid that they're derived from. And we'll talk about some specific examples as we kind of move along through this unit. Okay. So I am separating them out because that amino acid is going to really determine what their behavior is like, whether they act like a hydrophilic protein or whether they act like a hydrophobic lipid. It just kind of depends. All right? Okay. So let's take a look at some slides that I have for you guys and look at some of these chemical classes of hormones, and then we'll talk a little bit about how they work. Okay. I'm so happy I can control the lights from the wall. That's just like a Christmas gift. All right. So this is a diagram showing a couple of our representative steroid hormones that we have. The three that we have here, we have two hormones that come from an endocrine gland called the adrenal cortex. On Wednesday, we had talked about the inner part of the adrenal gland a little bit. That's the adrenal medulla. The outer part is the adrenal cortex. We will be talking about that in a lot of detail over the next couple of days. So don't feel like you have to understand everything about the adrenal cortex right now, but I'm just giving you some examples. A couple of hormones that come from the adrenal cortex are a hormone called aldosterone and a hormone called cortisol. And then we also have the ovary here as an example. One of the hormones it produces is called estradiol. The ovaries produce a whole family of hormones called the estrogens. And estradiol is one of the estrogens. All three of these are steroid hormones, though. And you can see how they're derived from cholesterol. So this is what a molecule of cholesterol looks like. You don't have to memorize its structure for the exam or anything like that. Don't worry about that. But you can see that all three of these examples, aldosterone, cortisol, and estradiol, all look very, very similarly to cholesterol. Okay? So you can see how they're derived from that sort of parent molecule. Cholesterol is a lipid. It is hydrophobic. And remember, the flip side of being hydrophobic is that we say that you're lipophilic, right? You don't like water, but you do dissolve well in lipids. Okay. We'll talk about the consequences of that here in just a couple of minutes. All right. Any questions about those? Again, we're going to go through all of these glands and all of these hormones in quite a bit of detail over the next couple of days, mostly in lab. But for now, I'm just kind of want you to understand that these all have a very similar structure to that of cholesterol because they are derived from cholesterol. This is another example kind of showing a family tree of some steroids. Again, we're not really worried about memorizing the table or any of the enzymes or anything like that are here. I just want you to see that you guys can come up with just about any steroid from cholesterol. And that's what we see happening here. Okay, let's talk for a minute about protein hormones. Okay, so proteins have an interesting sort of way in which they're made. Which really isn't unlike other proteins in our body. So there's really nothing special about them, but I just kind of wanted to sort of talk about them a little bit. So protein hormones are proteins And so the genes for them are found in our DNA. We see that transcription and translation are happening just like they would for any other protein. But one of the things that we learned when we learned about protein synthesis in Bio 1 is we learned that when that protein is made, it often has additional amino acid sequences on it, right? So if this amino acid chain right here is my hormone, right? This is my hormone right here. It hasn't been folded yet, but this is that chain of amino acids. Pretend that this is a chain of several hundred amino acids that we have right here. There's going to be additional amino acid sequences added onto it that are going to serve a number of purposes, right? So I might have an additional amino acid sequence right there. And in the case of hormones, I have a couple of additional amino acid sequences. So I have a little bit kind of tacked on right here. Okay? With those additional amino acid sequences that have been added on, we call this a pre-pro hormone. Okay? We call this a pre-pro hormone. You guys remember what did you talk about Bio 1? What the purpose of those additional amino acid chains are? There's a couple of different things they can do, but anything? Bio 1 was like a lifetime ago, right? Yeah.
I'm just guessing, but like, is it to protect the sequence here?
Is it protect? Is it protection? Is it to protect the hormone? Okay, that's a good guess. I don't know if they necessarily have a protective function, but that's definitely a good guess. So, where do you guys remember where proteins go after they leave the ribosome? It's the Golgi body, yeah, right? It's the Golgi apparatus. Okay. If you're a fan of the rock band Fish, they've got a whole song about the Golgi apparatus. So they'll remind you about that if you like. It's called Golgi apparatus. So at any rate, yeah, they usually have to go to the Golgi apparatus, right? The Golgi apparatus is going to do a number of things, but one of the things that it does is it will package those proteins into vesicles so that they can be secreted from the cell. Okay? How does that amino acid chain know that it's got to go to the Golgi apparatus? Well, it's got an amino acid sequence directing it there. Okay? So these additional amino acid sequences very often are just a way for us to direct this thing around the cell so that it can be ready to get shipped out, right? It's kind of like a barcode in a warehouse, right? How do we know which truck to put that on? Well, it's got a specific barcode on it, and it's going to direct it towards the truck that it needs to go on for delivery, right? Same idea here. We just are using amino acid sequences to do that. Okay. So these amino acid sequences are largely sort of directing, these guys. So what happens is once we get to the Golgi apparatus, we can usually cleave off at least part of that amino acid sequence because, hey, we got to where we need to go, right? When we cleave off that amino acid sequence, that becomes a pro-hormone. Okay? So the name changes. I guess you could call that first amino acid sequence the pre, right? And now this next one is the pro. Okay? So this is the pro-hormone that we have right here. When these additional amino acid sequences are on that hormone, this is largely inactive at this point. Remember that the hormone itself will fold into a really specific three-dimensional shape. And that shape is important because it has to fit perfectly into its receptor. Okay? With that additional amino acid sequence on there, it's not going to fold into the right shape. It wouldn't be able to fit into its receptor. So if you were to take a pre-pro hormone or a pro hormone and dump it into the bloodstream, it's not going to probably do anything at all. Okay? It's just going to float around and it's not going to be able to fly the receptors. Okay? But nevertheless, that that initial amino acid sequence is going to be divided to the Golgi apparatus so that it can get packaged into secretory vesicles so that it can get ready to dump into the bloodstream. Okay. Once it gets packaged into the secretory vesicles, what we do is we cleave off that additional amino acid sequence, and it becomes an activated hormone. So that when those vesicles are secreted by exocytosis, we have an active hormone that's now being secreted into the blood. So there are a few steps involved in protein's hormone synthesis, but they're really not unlike what we might see for any other protein in the body. So there's not a whole lot of difference there. And then amine hormones are a little bit variable. Let's talk a little bit about amine hormones. Come on up over here. When the screen is down, it really limits what I can write on the board here. Gotta use my resources. So the amine hormones here. There's two primary amino acids that we use for the amines. The two primary amino acids that we see is the amino acid tryptophan and the amino acid tyrosine. One of the most common hormones derived, or one of the probably the ones that we're the most familiar with, derived from tryptophan is the hormone melatonin. Right, it's the hormone melatonin. We talked a little bit the other day about epinephrine and norepinephrine. Epinephrine and norepinephrine are derived from amino acid tyrosine. Remember we said they're the catecholamines, right? Catecholamines. Call them that long. Those are our catecholamines that are derived from tyrosine. Another one that's derived from tyrosine is thyroid hormone. We'll talk about that probably today in lab. We'll talk about thyroid hormone today in lab. So, a couple of examples of hormones derived from different amino acids. Wouldn't expect you guys to know what all of those do yet. We haven't talked about them in a lot of detail. But you probably know about melatonin. What does melatonin do to us?
Just make us sleepy.
It's the sleepy time one, right? Yeah, melatonin is really important in regulating essentially our sleep and wake cycle, right? Again, we'll talk a little bit more about it when we talk about the pineal gland, probably next week in lab. But yeah, it's the one that kind of makes us go to sleep. There's this kind of urban myth that the amino acid tyrosine makes you sleepy, right? It makes you go to sleep. And there's also an urban myth that a food that's particularly high in tryptophan is turkey, right? So there's this whole idea that you get sleepy after Thanksgiving dinner because you ate a lot of turkey, you ate a lot of tryptophan, and now you're going to sleep. You guys are all probably way too young to remember it, but there was an episode of the TV show Seinfeld centered around this whole premise where they were trying to overdose one of George's girlfriends on tryptophan to make her go to sleep so they could play with her vintage toy collection. It's a really stupid episode. It was a really stupid show, but that's kind of the urban myth that we have, right? The reality is that turkey is jam-packed full of other amino acids as well. I mean, it's meat. So there's all sorts of other amino acids. There's not an abnormal amount of tryptophan in turkey, at least not as I understand it. And the reason that you get sleepy after Thanksgiving dinner is not because you ate turkey, it's because you ate a lot of food. And your gut is now full and your belly is now happy and now it's sleepy time. So you're going to sleep for a completely different reason after Thanksgiving meal. But at any rate, it's kind of an urban myth that sort of perpetuates itself around tryptophan. But I think that myth comes from the fact that melatonin is derived from tryptophan. Melatonin makes you sleepy, tryptophan does not. So anyway. So we have three different chemical classes of hormones. We have the proteins, we have the steroids, and then we have the amines. Are there any questions so far? Not yet? Sweet. All right. Let's go ahead and talk a little bit about how these hormones work then. So to understand how these hormones work, we have to understand where they bind to their receptor. Again, in order for any hormone to work, regardless of its chemical nature, it has to bind to a receptor. Okay? Remember, these hormones are going everywhere in the body. Right? And as a result of that, we have to have something that's going to make certain cells respond to that hormone, and then in the absence of that receptor, those other cells are going to ignore that hormone, right? To that end, it's not really unlike making a cell phone call. Right? If I wanted to call somebody on my phone right now, I could pick that phone up and I could dial a number and I could hit send. But the way that cell phones work is that message goes everywhere, right? When I hit send and I'm making that call, that message is going to go to every single one of your phones. It's going to go across Magnolia. It's going to go to everywhere around, right? It's going to hit repeaters, and that message is going to get sent even farther. But at the end of the day, when I call a number on my phone, there's only one phone that rings, right? And that's the number of the phone that I dialed, right? And the idea is that phone, the number that I dialed, that phone number is the receptor, okay? And that phone is the only one that responded to it. All of your phones are going to ignore that signal because they don't have the receptor, the number that I dialed, right? So hormones are the same way. That message goes everywhere, but if the receptor is there, the cell will respond to it. If the receptor is not there, the cell will ignore it. Right? So let's go ahead and take a look at this. So just make a quick note to ourselves, because the vocabulary can be a little bit confusing at times. Right? Let's remind ourselves that proteins are hydrophilic. Remember, that means water loving. But as kind of a corollary of that, if they dissolve well in water, they do not dissolve well. They do not dissolve well in fats and lipids, right? So they are hydrophilic, but they're lipophobic. Steroids, we're going to ignore amines for the time being, but steroids are hydrophobic, but they are lipophilic. Hydrophobic, but they are lipophilic. Okay? And this is an important thing to remember. I'm focusing more on the lipid side of things, whether or not this is lipophobic or lipophilic, because we have to contend with the cell membrane, right? On that target tissue, it is made up of cells, just like every other thing in your body, and it has a cell membrane, a plasma membrane. And we know that that plasma membrane has a fatty core, right? That's made up of those phospholipids. So we have to contend with that plasma membrane and the fatty core of it. So this ultimately becomes more important for how a hormone is going to work than the other one. We'll talk about some aspects of the other one a little bit later, but for now, let's focus on whether or not that thing can cross the cell membrane. Okay? Protein hormones are unable to cross the cell membrane. Right? So what that means is that they have to bind to a receptor on the outside of the cell. Okay? So if we were to make a table here. Proteins, and we have steroids. Okay? Proteins have to bind to a receptor on the outside of the cell membrane. extra meaning outside of, right? So proteins bind to extracellular receptors. And in the diagram that I have projected for you guys, in the upper left, you can see what that extracellular receptor might look like. You can see that it's a protein very often embedded in the cell membrane right here, but the receptor, the actual open area of that, is located outside of the cell, so that protein hormone can bind to that receptor. Okay. Now, because it's binding to a receptor outside of the cell, that means that we're going to have some sort of a process to get the message inside of the cell, right? So it's binding to a receptor here. How are we going to transmit that message inside of the cell? This usually is going to involve employing some sort of a molecule that we call a second messenger. Okay? In this case, the hormone is the first messenger. It's bringing the message to the cell. Because it can't cross the cell membrane, we have to have something inside of the cell that will continue to send that message. Does that make sense? So we call those molecules second messengers. So we're going to have to use second messengers. We're not going to go too far down the rabbit hole of listing all of our second messengers and the way that they work. You might have heard of the molecules like cyclic AMP, CAMP. Glycerate, another one. We have G protein coupled receptors. We have all sorts of different things that we use. But they're always going to use some sort of second messenger to get the message across inside of the cell. Okay? Again, we're not going to worry about the individual ones. I'll write cyclic AMP up here as an example. Okay, so as an example, we have cyclic AMP. AMP in this case stands for adenosine monophosphate. But there's a whole bunch of others, and if we had the time, or if this was an endocrinology class, we would obviously go into the way that every single one of them works, but we don't have that kind of time in this class. So we'll just generalize and give you a single example of a fairly common one that we see. Okay. So protein hormones bind these extracellular receptors. They employ a second messenger to get their message inside of the cell. And then what they do once they get that message into the cell is the typical cellular response is to modify the activities of proteins that already exist in the cell. Okay? So they're modifying proteins that already exist inside of the cell. This could mean turning a protein on, it could mean turning a protein off. It could mean amplifying the activity of a protein that already exists to make the cell do more of that thing. It could mean turning that protein down to make the cell do less of the thing that it does, right? But they're modifying proteins that already exist in the cell. Okay. Let's try that differently. Let's just write modify existing proteins. Modify existing proteins within the cell. As a result of that, because those proteins are already there, they just have to be modified in some way. Protein hormones typically work pretty quickly. Once they're released, they typically work pretty quickly. And as a result of that, we see pretty rapid physiological responses to protein hormones. On the fast end of things, we can see or measure physiological responses to protein hormones in milliseconds. Okay? On the really long end of things, we're looking at minutes. But nevertheless, we're looking at pretty fast responses to protein hormones. Okay? Body responds really, really well to them because we don't have to make new proteins. We just have to modify proteins that are already there. Maybe you yourself have experienced this, or maybe you know somebody who has to take insulin to help to regulate blood glucose levels. But next time that happens, pay attention from the time that person may take their insulin to the time that you notice a behavior change or the way that they feel. It doesn't take long at all. I mean, we're talking just a couple of minutes, and you can see a really profound difference in the way that they act and probably the way that they feel, because their blood glucose changes so rapidly in response to the insulin that they take. Insulin is a really, really good example of a protein hormone that we'll be talking about in lab probably next week. Okay. So, at any rate, really fast responses to protein hormones. And again, these are probably the most common proteins or hormones inside of our body. See if we can hustle through steroids really quickly. So, steroids are our lipophilic signaling molecules. And because they are lipophilic, it means that they can cross some membranes really, really easily. So the receptors for steroid hormones are not found extracellularly, they're found inside of the cell. Where inside of the cell kind of depends on the individual circumstance. Sometimes the receptor's floating around in the cytoplasm, sometimes it's found inside of the nucleus, but nevertheless, we're looking at intracellular receptors. What will happen is that once that hormone binds to its intracellular receptor, we see a couple of different examples right here. This is a cytosolic receptor. This is a nuclear receptor right here. But once that hormone binds to its receptor, that what we call hormone receptor complex will act as a transcription factor for the production of new proteins. So what we'll start to do is we'll start to make new proteins in response to this hormone receptor complex. Okay. So we'll say here that the hormone receptor complex acts as a transcription factor. And that leads to new proteins. Okay. That leads to new proteins being produced. So protein hormones were acting on proteins that already existed in the cell, right? These guys were acting on proteins that already existed in the cell. These guys are making new proteins. That takes a lot longer. It's not an instantaneous response. So it might take, on the short end of things, we might see responses in a few minutes, but usually in most cases we're looking at hours, or in some cases even days, before we see measured responses from the cells. Okay? So this is a lot slower of a process when you have to make proteins. So steroid hormones are not hormones that we see this instantaneous response in our body, right? It's long-term, a little bit more sort of an invested response that we see from our body. Okay? So it's a lot longer. Like I said here, minutes, or in some cases even days. All right? Okay. Okay. So I think that puts us at a good stopping point. What we'll do in lab is we might talk a little bit more theory, we'll kind of see, but then we'll start to actually go through some of the endocrine glands, start to look at the hormones that they produce, and then we'll kind of go from there. All right? All right.